All right, welcome to the third part of Chapter 8, Patterns of Inheritance, where we want to look at extensions of the laws of inheritance. See, up to now we've been looking at Mendelian genetics, which are fairly simple. You've got a dominant or a recessive allele, but a lot of times there are exceptions to that. One of these is incomplete dominance. And in incomplete dominance, the dominant allele is not fully dominant over the other. And so what ends up happening is if you have a heterozygous genotype, the phenotype is going to be somewhere in between the two homozygous phenotypes. And in snapdragons, if we cross a red, which is dominant, to a white flower snapdragon, the heterozygous condition gives us a purple color, or a pink. I guess that's a pink color. To me, it looks a little more purple than pink, but they're saying it's pink, so it's pink. Okay, but it's in between the two. It's kind of an intermediate. We see this in guinea pigs with white and brown guinea pigs. We get a cream color. There are other instances of incomplete dominance. In codominance, both alleles are fully expressed in a heterozygous individual. And the example of this is the ABO blood groups in humans. If you're A type A blood, then you produce red blood corpuscles that have a specific carbohydrate on their surface. Okay, if you're type B, your red blood corpuscles have a different carbohydrate on their surface. Okay, so members of group A that have this carbohydrate produce antibodies against type B. And so if you mix type B with an A blood person, their antibodies are going to bind to this and cause clumping in the bloodstream, and this can be life-threatening. So you can't mix B with A in a person who's A, and you can't mix A with B in a person with B because type B has antibodies against type A. And so you add these to this person, and these antibodies are going to cause clumping. Okay? If you're AB, you've got both carbohydrate types on the surface of your red blood cells. So you don't have any antibodies in your plasma. And so type AB can get blood from either A or B. Okay. If you're type O, you've got to be careful because if you've got both the anti-A and the anti-B antibodies, and so you get any blood from any of these is going to cause clumping. Okay. But that's the variations we see in ABO blood groups. And so this is showing you possible matches now you notice O negative is the universal donor because the recipients, all of these recipients can get blood from O negative. O positive, only AB positive, A positive, B positive, and O positive can get blood there. You notice that AB positive can get blood from anybody, right? And so this is why they blood type you for both your blood type and the Rh factor before they determine whether or not they should give you a transfusion of blood. Epistasis, this used to be called polygenic inheritance, is that some traits, there are multiple genes and the products of those genes that contribute to the trait. One of these is human skin color. There are more than 100 different gene products that determine how much melanin you produce and where it's deposited. And it's the melanin in your skin that causes your skin color from the very light to the very dark. Okay? And there's a huge range of variations because there's so many different gene products that there's a different ratios of how these can affect each other. And that's again all because of independent assortment. We can't control that. Pleiotropy is the opposite of that. This is where one gene influences more than one trait. There are several examples of this that are diseases. Phenylketonuria, which is your inability to metabolize phenylalanine. Phenylalanine is the amino acid that's a precursor for NutraSweet, which is an artificial sweetener used in a lot of diet beverages and diet foods. But there are people who can't break this down. And so they have to make sure they keep this out of their diet, because if they were to have it heavy in their diet, it leads to intellectual disability, seizures, behavioral problems, mental disorders, a whole slew of problems. Sickle cell anemia, cystic fibrosis, Marfan syndrome are all syndromes associated with genetic syndromes associated with pleiotrophy. There are also complex variations in traits. Not all the proteins that a gene codes for are going to be identical because of mutations. They might be slightly different, so you might get a slightly different gene product in one individual over another, and not all mutations are bad. It can cause a beneficial trait too. Remember, there are interactions among genes, and the environment contributes too. So they've shown this in studies with twins, where they've taken identical twins and raised them in very different situations, and they've developed slightly differently from each other. This can change steps in a metabolic pathway, and you've seen enough metabolic pathways to realize how complex they can be, and this can lead to a variation in phenotypes. Take the snowshoe hare. Okay, we live in Michigan. You get up north, we've got snowshoe hares, and in the summer the hare's fur tends to be brown, and yet in the winter its fur turns white. And the reason for that is because by changing, it allows it seasonally appropriate camouflage. and this reminds me of an instance when I was in high school. I had a friend who had a cabin up north, and we went up on spring break to go hunting, and we were hunting snowshoe hares. And that particular year, we had an early melt, and the hares were still white like this guy is, but the background was more browns because of there was no snow. And I was walking through, and I looked over, and there was a stump, and there was a snowshoe hare standing really still in front of that stump, thinking, you can't see me because it's winter. And I'm like, dude, you stand out like a sore thumb. And I would like to tell you that I didn't take advantage of the situation and let him go, but we had snowshoe hare for dinner that night. Water fleas. Okay, some of you may have seen water fleas in your pond water samples. Normally they look like this. They're shorter and they're a little longer and round. Okay, but if they're in a pond with a lot of predatory insects that feed on them, the chemicals emitted by the predatory insects will cause them to grow a longer spine and a pointy head, which probably makes them harder to consume than these are. All right. Now, when you have variation in a continuous variation like in skin color, you're going to have a lot of people in the middle and fewer people at the extremes, and this is going to end up forming what we call a bell curve. Okay, very few individuals at the extreme. So if we're talking skin color, very few really, really dark, very few really, really light, and most people somewhere in this middle range. Okay, that's what we normally see in continuous variation, and we can see this in eye color. Eye color is another phenotypic trait that's controlled by multiple genes, and there's a variety of eye colors from really, really dark to really, really light, and we see that same variation. Now, when we have genetic variation, there are over 6,000 documented abnormalities and disorders. Now, an abnormality just means an uncommon version of a gene. There's no medical problems associated with it. A genetic disorder is heritable, meaning you pass it on or have the potential to pass it on. And this can be a major medical problem, sometimes mild, but sometimes it can be a big deal. If we look at dominant disorders where you only need one gene, one dominant allele, achondroplasia dwarfism is one of the forms of dwarfism. That's probably the most common form of dwarfism. The individuals are normal except for their dimensions. Their trunk is much shorter, their limbs are much shorter, but other than that, they're very much normal. Aniridia is a situation where you don't form an iris in your eye. Without an iris, you can't control how much light comes into your eye. And so it's a very, very painful condition. People with this have to wear really dark glasses all the time because any light is going to be painful for them. Camptodactyl is when you have rigid bent fingers, makes very limited dexterity. Huntington's disease is when you have a degeneration of your neuromuscular system. And this usually kicks in when you're in your early 30s to late 30s after you've likely already had children and possibly passed it on. So someone who has a parent with Huntington's disease, their children have a 50% chance of getting it too because they don't know which gene their parents passed on. Marfan syndrome affects connective tissue, especially the connective tissue that holds the various regions of the heart and the tissues in the heart together. And so people with Marfan syndrome are often likely to have cardiovascular issues at a young age. Polydactyl means you've got extra fingers, toes, or both. This is not uncommon, and it's certainly not an issue other than you have extra fingers and toes. We see this a lot in cats. My dad was born with 6 fingers on each hand, and they clipped the sixth one off when he was little. Progeria is drastic premature aging. And when you see people with progeria, these are kids in their single-digit years, 8, 9 years old, and they look like little old people. And they die very young because their body is aging way, way faster than it should. If you have a dominant allele that's lethal and it would kill you right away, it's probably going to kill you before you're able to have children and possibly pass that on. And so those kind of things. And you're saying, well, how come progeria doesn't these children die before they ever had children, so why does this persist? Well, these can also be formed by a spontaneous mutation in the gene that controls this. And that could happen at any time. It doesn't have to be passed on. Recessive disorders, you've got to get the recessive allele from both parents to end up with a disorder. Okay? So if the parents so albinism is the absence of pigmentation. Very light skin, pinkish red eyes. You've got to be very careful in the sun so you don't get sunburned too much. But the parents could be normally pigmented, but they would both be carriers because they each passed on a recessive allele. Cystic fibrosis occurs when, so you produce mucus in your lungs to trap material that you breathe in, dust and bacteria and all kinds of pollen grains, things like that. And it gets caught in the mucus in your lungs, and then cilia sweep it up and out because the surface of the cells secrete a protein that makes them slippery. People with cystic fibrosis have an abnormal protein that isn't slippery on those cells, and so they have trouble getting rid of the mucus. And as they accumulate that excess mucus, bacteria thrive in it. They're prone to severe bacterial infections in their lungs, and without significant medical treatment, they're probably going to die in their late teens, early 20s. Ellis Van Creveld is another that causes a type of dwarfism with heart defects. Friedreich's ataxia messes with your ability to move, motor and sensory function. Phenylketonuria, we already talked about, can't break down phenylalanine. Sickle cell anemia is when your red blood corpuscles aren't flexible and pliable, but they have a rigid sickled shape. And then Tay-Sachs disease, which is related to abnormal lysosomes, which can lead to early death and deterioration of mental and physical abilities. Because these require the parents to both contribute a recessive allele, the more a population inbreeds, the more likely that the increase of these disorders will happen. There are also some disorders that are X-linked, which means they're on the X chromosome. And the significance of this is if you're a chromosomal male that is XY, has one X chromosome, one Y chromosome, then if you have a trait, you have a gene on the X chromosome, it's going to be like a dominant allele because you only have one X chromosome. So whatever's on it's going to be expressed. If you're chromosomally female with 2 X chromosomes, one of your X chromosome is inactivated and becomes something called a bar body. And when it's inactivated, those genes aren't expressed. And so if whatever the disorder is on, the gene is on the X chromosome that's been inactivated, you're not going to pass it on, and you're not going to, it's not going to be an issue. If a male has it, it's going to be an issue. This includes things like androgen insensitivity. So look here, an XY individual has the traits of a female, and they're sterile. Now, think about what's going on in society and all the issues about gender. If you're XY, you're male, and if you're XY, you're female. Well, here's an XY that has the traits of a female. All right. Red-green color blindness. This is going to affect guys more than it does girls. Hemophilia, which you're not able to clot as well, and so you got to be careful about injuring yourself so you don't bleed excessively. Various muscular dystrophies, where you lose loss of your muscle function. All of these are going to be, we're going to see these in society more in men than in women because of the X chromosome inactivation. Drosophila are fruit flies, and the gene for eye color is on the X chromosome. Red eye color is wild type and dominant to white. And so when we see red eye type, if these are both males, and this is on the X chromosome, then we got to remember in insects, it's not XY like it is in humans. So this is probably not a good example to have here. Remember non-disjunction? When in meiosis, we didn't fully separate the sister chromatids, either in meiosis 1 or meiosis 2. And so we had aneuploidy occur, where individuals had either too many or too few copies of a chromosome. Instead of being, diploid, they had one too many, and they had trisomy, or one too few monosomy. The most common of these is trisomy 21, which causes Down syndrome. Again, non-disjunction for the sister chromatids to fully separate. There's also Turner syndrome, where a chromosomal female is lacking the second X chromosome. There's Klinefelter syndrome, where you're XXY. There's triple X syndrome. There's XYY syndrome. Now, a couple of these are very mild, not much issue with it. But, typically we say, well, if you're XX, you're female, and if you're XY, you're male. But look, what do you do with these two? What's this person? So sometimes, chromosomal gender isn't always the same as biological gender. And there's a lot more to it than we can oversimplify issues. Down syndrome, extra 21st chromosome. Extra 21st chromosome, we can see this in a karyotype. Remember, a karyotype is a photographic inventory of the sister chromatids that are taken during metaphase of mitosis. Okay, and here we can see there's the extra 21st chromosome, and this is what causes all the characteristics associated with Down syndrome. Of course, we've seen that before. Okay, now we can genetically screen, and this lets you know what genes you have. This lets you know what genes you have. Why bother screening? Well, I'll give you for instance. My grandmother on my father's mother died of Huntington's disease when I was a kid. And when that happened, we knew at that point Dad had a 50% chance of having the gene. If he ended up having Huntington's disease, then I would have a 50% chance of having it. And so, for a long time there was a lot of, nobody knew. And then they developed a test to screen for it. And so Dad decided he was going to go in and get tested. And he had to have counseling before he got tested, and they tested him. He had counseling after the testing because they didn't want him to do anything rash if he got results he wasn't happy with. And then we found out that he didn't have the dominant allele for Huntington's disease. He was never going to get it, okay? Which immediately meant my brother and my sister and I weren't going to get it from him. It was not an issue anymore. But for a long time when we were young growing up, we knew about it and we thought about it. And that determined a lot of my behaviors, when I was younger. I lived my life like a rock star for a long time because I thought, if Dad gets it, I get it, then I don't want to die young with regrets of not having fun. So I probably had too much fun. But there are a lot of reasons why people might want to know what genes they have. There could be a problem with that, though. If they did a genetic screening and they found that you had genes that were associated with addiction, you were more prone to addiction, and a potential employer got their hands on that, could that keep you from getting hired? If an insurance agent got it, could that keep you from getting health insurance? So, places like the American Civil Liberties Union, the ACLU, doesn't really like the idea of a lot of genetic screening to identify what people might or might have. Another reason you might want to diagnose is you might want to know what the risk was of you passing something on to your child. Maybe, cystic fibrosis, there's a significant number of carriers in our population. If you were with somebody and there was a history of cystic fibrosis in your family, you might want to know if you were a carrier or your possible significant other was a carrier, because then you would have a 25% chance of having a child with cystic fibrosis. It might impact your decision whether to have children. I have a friend who developed multiple sclerosis, which is a recessive disorder, when he was a young man. And him and his wife made a decision not to have children because he didn't want to run the risk of passing it on. There's also possibly prenatal diagnosis. Do we want to check a developing fetus to see if there's anything wrong? Now, there's non-invasive ways to do this with, ultrasound, sonographies, where you don't go into the womb. But then there are invasive methods of this too, where you actually go into the womb next to the developing fetus. In amniocentesis, what you're doing is you're going into the amniotic fluid, which is right around the fetus in the placenta, and as the fetus is moving, it's scraping off cells, and some of those are going to be in metaphase. So you go in and you suck out some of that fluid, you find a cell in metaphase, you do a karyotype of the chromosomes, and you look to see what's there. Chorionic villi sampling is a little less invasive. The chorionic villi are like finger-like projections from the placenta, so it's a little farther away from the fetus, but you're still invasive into that area. A fetoscopy is when you actually take a camera in there and video the developing fetus. And so the problem with these invasive techniques is, what if you accidentally introduce bacteria and cause an infection? What if you accidentally hit the developing fetus and cause damage? And so, whether you choose to do these things or not, and this was one of your discussion questions, this is going to vary from person to person. And it's certainly there may be situations where some of these are warranted, and there may be situations where none of them are warranted. And I think probably it's a very deeply personal decision. This is a conventional ultrasound, right? And see these on Facebook all the time. And I look at this and I say, okay, here's the beak, here's the eye, there's the head, there's part of a wing. How'd the bird get in there, right? Okay. And then this is a 4D ultrasound, which shows a little more detail. And then this is a fetoscopy. This is when you actually take a camera in and you take a look at the developing fetus. Here's a fetus developing within the amniotic sac. Okay, amniotic fluid's in here. So in amniocentesis, you're going into this to try to get some of the fluid that might have cells that this fetus has sloughed off. Okay, here are the chorionic villi. This is going into here. You're likely that any cells that are in this have made their way to here. It's less invasive, but it's still fairly invasive. So that's the end of this last part of Chapter 9, or Chapter 8, excuse me. Next time we'll start Chapter 9, Molecular Biology. We'll look at the structure and replication of DNA.